Who's ready for some fun with biomolecules? All right, we're going to move into the rainbows portion of our unit. So the sunshine part was the chemistry, and now we're going to use biomolecules to go into the rainbows part. All right, first thing, the word organic. Contrary to what you hear in the media and the news and the sales and the marketing people and whatever, in chemistry, organic just means carbon-based. So that just means we're talking about molecules that have carbon at their heart, at their core. So a molecule based in carbon is an organic molecule. It has nothing to do with pesticides or GMOs or any of that other stuff that you might hear. You can get organic stuff anywhere you want. You don't have to go to Whole Foods for it. So since that's our definition, is a compound based on carbon, here are some organic molecules. Sugar is an organic molecule, fat is an organic molecule, starches are organic, proteins are organic, okay? Well, then what's not organic? Well, first of all, our word for not organic is inorganic, because in is the prefix that means not. So inorganic, some inorganic molecules are things like water, H2O, there's no carbon in that. Oxygen, O2, there's no carbon in that. Minerals are inorganic. Vitamins are usually inorganic. Fun fact, carbon dioxide is a little tricky. It's one that it depends on who you ask. So carbon dioxide, obviously, duh, has carbon in it, right? So you're like, yeah, that's totally a carbon-based molecule, duh. But carbon dioxide is a really tiny little molecule, and carbon dioxide doesn't really behave the way the rest of the organic molecules do. And so a lot of people will tell you that carbon dioxide is inorganic, although strictly, according to the definition, it is organic. I don't know, it's just one of those things. All right, so the chemistry of carbon. So carbon itself has some really unique qualities, and that's why it's so good as this basis of life, this basis of living things and anything that's alive on our planet. Carbon can actually bond with many other elements. It can make up to 4 bonds. So if you remember when we were doing that molecules lab, when you were modeling with the molecules in the lab, you had those little black molecules and there were 4 different holes because you can make 4 different bonds. So some of its best friends are hydrogen, oxygen, phosphorus, nitrogen, sulfur. Those are all different things that really like to bond with carbon, that do really well with carbon. So if you think about it, you have a skeleton in your body that holds up your skin and your organs. And so we talk about a carbon skeleton or a carbon backbone that holds together organic molecules. So oftentimes when you're looking at an organic molecule, you'll look at a long chain of carbons. And we call that a carbon skeleton or a carbon backbone, because carbon bonds with other carbons. So you can go carbon, 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 and then have a whole bunch of stuff coming off the sides. So in this example of the saturated fatty acid, you have this carbon backbone going down the center, and then you have all these hydrogens on the outside, and then you have this carboxyl group on the end. So you have that you have a double bond here, and then you have a bond to the oxygen, and then this oxygen is bonded to this hydrogen. So that's what this carboxyl group is. It's a C double bonded to O, and then the hydroxide group. And then the carbon is bonded to this other carbon. So 1, 2, 3, 4. So that carbon has 4 bonds. The oxygen has 2 bonds, 1, 2, and then this oxygen has 2 bonds, 1, 2. But what's important here is to look at the carbon backbone. So we have this nice carbon skeleton that's holding our molecule together. You can get a bunch of different backbones. You can get just 1 carbon in the middle. You can have 2 carbons together. You can have a bunch of carbons with some double bonds, some single bonds. You can get rings. You can get branched structures. You can do all kinds of different things with carbon because carbon is so cool. All right, so the word macro means really, really big, right? So a macromolecule is just a giant molecule. So we talk about macromolecules. We're talking about really, really big molecules. So we're no longer talking about CO2 with 3 atoms in it. We're not talking about even, really glucose with 24 atoms in it. We're talking about molecules that have thousands and thousands and thousands of atoms in them. Okay, so they're made up of smaller molecules and then atoms within those smaller molecules. So here's how macromolecules work. We have monomers. Mono means one, mer means piece. So a monomer is 1 piece. And so I have some different monomers that are here all over the place. So there are your monomers. Poly means many, mer means piece. So a polymer is many pieces. So if you put a bunch of monomers together, you end up with a polymer. So monomer plus monomer plus monomer plus monomer plus monomer gives us polymer. Yay! Okay, so here it is again. Monomer plus monomer plus monomer gives us a polymer. That process, one thing we can call that is polymerization. Polymerization. We're just making a polymer. Okay, but really when we talk about it in terms of organic molecules, we're talking about dehydration synthesis. So with organic molecules, dehydration synthesis is how we take monomers and make them a polymer. D means take away, hydration refers to water, synthesis means you're putting something together. So dehydration synthesis means we're putting something together by taking out water. So if you take a monomer and you attach it to another monomer, a water molecule is going to come out. If you attach another monomer, a water molecule comes out. Attach another monomer, a water molecule comes out. Attach another monomer, a water molecule comes out. Now notice not all monomers are the same, and that's okay. But every time you attach two monomers, a water molecule is going to come out. That's why it's called dehydration. We're taking water out, synthesis, while we're building these things together. Okay? The opposite of dehydration synthesis is hydrolysis. Hydro, again, refers to water. Lysis means breaking. So hydrolysis means breaking apart using water. So we're using that water to break something apart. So we've got a polymer, just add water, and we're going to break a monomer off. Just add water again, we'll break another monomer off. Add water again, break a monomer off, add water again, break a monomer off. So hydrolysis is the act of using water, adding water, to break these polymers apart into their monomers. All right, so we have 4 classes of biomolecules, and biomolecules are we're just talking about macromolecules here that are biological macromolecules. So I'm going to use the word macromolecule and biomolecule interchangeably here because I only care about the ones that are biological. It's a bioclass. So there are 4 classes. You have to know these, so you need to memorize this. I'm going to be asking you from now until the end of the year, what are the 4 classes of biomolecules? You've got to know them. Carbohydrates, lipids, proteins, nucleic acids. Carbohydrates, lipids, proteins, nucleic acids. Okay, so each one of these is a polymer. Each one of these is made up of smaller pieces called what? Monomers. Okay, so the monomers of carbohydrates are called monosaccharides. The monomers of lipids are fatty acids and glycerol, and that needs an asterisk on it because lipids are a little funky, but in terms of memorization purposes, fatty acids and glycerol is good enough for me. The monomers of proteins are amino acids, and the monomers of nucleic acids are called nucleotides. Again, you have to know this. Memorize it now, memorize it often. You got to know it all the way through the end of the year. It's just one of those things that goes all the way through pretty much every unit we're going to talk about. Okay, carbohydrates specifically. The monomers of carbohydrates are called monosaccharides. Mono means one, saccharide means sugar, so one sugar. It's a simple sugar. So glucose is an example of a simple sugar. But then you can take glucose and attach to glucose and attach to glucose and attach to glucose, and you can get a much bigger carbohydrate. So some examples of really complex carbohydrates are starch, like in your pastas and in your breads and in your potatoes. Cellulose, which is what we often refer to as fiber. Plants use it in their cell walls, and it's not really digestible for us. And so, like lettuce, when you're eating lettuce, you're eating a lot of cellulose. When you're eating spinach, you're eating a lot of cellulose. Chitin is the stuff that bugs use to make up their exoskeleton, so that's what makes a bug crunchy. And then glycogen is a complex carbohydrate that you use inside your body to store energy. There are different types of simple sugars as well. There's glucose, there's fructose, there's galactose. So remember a few slides ago I showed you that some of your monomers were triangles and some of them were squares and some of them were circles and some of them were hexagons. That's because there are different monomers out there. Carbohydrates are used primarily for 2 things. They're used primarily for structure, so to build things, and for easy access energy storage. So we're going to build things with them and we're going to use it to store energy. So it's a quick, easy way to get our energy. So monosaccharide plus a monosaccharide, when we add them together, water comes out, dehydration synthesis. Okay. Now, if we've only got 2 of them, this is called a disaccharide because the prefix di means 2. So it's a disaccharide. But now if we add another monosaccharide on, and another, and another, now we have a polysaccharide. And an example of a polysaccharide, remember, would be starch, cellulose, chitin, glycogen, any of those guys. All right, lipids. Lipids have the same functions as carbohydrates, but slightly different. So they're used for structure. But this time when we're talking about energy storage, lipids are long-term energy storage. So lipids are kind of like the bank account that you go to get your energy out of, whereas, well, actually, no, that's not true. Lipids are kind of like your college fund, whereas carbohydrates are more like your bank account that your ATM card works in. So you can pull money out of that checking account or that savings account pretty quickly. Lipids are like your college fund. It's going to be really hard to get to that money. That's the way energy is with lipids. Really long-term energy storage. Lipids are made up of carbon, hydrogen, and oxygen, just like carbohydrates are made up of carbon, hydrogen, and oxygen. It's just that they're in different ratios. Lipids are what we call hydrophobic. Hydro, again, referring to water. Phobic, phobia, afraid. So they are water-fearing. They are water-fearing. So lipids are afraid of water. They do not want to be like water, be by water, and that's because they're nonpolar. Water is polar, lipids are nonpolar. They're just not going to mix. Generally, when we talk about the monomers of lipids, we're talking about glycerol and fatty acids. So here's a glycerol molecule right here. And then here's a fatty acid, here's another fatty acid, and then here's another fatty acid. So we have our glycerol and then 3 fatty acids, which is why this is called a triglyceride. The tri refers to the 3 fatty acids, and then the glycerol, the glyceride. So a triglyceride, 3 fatty acids. We can have saturated and unsaturated fatty acids. If you think about what saturated means, saturated, if we're talking about a sponge, would mean that it's so full of water that absolutely no more water could possibly come out of it or could possibly go into it, and that's a saturated sponge. If we talk about an unsaturated sponge, that means that we could get more water into it. It's not completely full. So there's more there. That's how fatty acids work too. So a saturated fatty acid has a hydrogen absolutely every single place there could be a hydrogen. So any place you could attach a hydrogen on this carbon skeleton, you have a hydrogen attached. But an unsaturated fatty acid does not have that happen. So here we have this double bonded carbon. Now, if we could pop this double bond and this guy made another bond and this guy made another bond, we could have a hydrogen here, we could have a hydrogen here, and then we'd have a saturated fatty acid. But that's just not the way this particular fatty acid is built, and so it's an unsaturated fatty acid. We can have triglycerides as a type of lipid. We can have phospholipids. We can have steroids. We can have cholesterol. There are a bunch of different forms of lipids. Proteins. Excuse me. Proteins do almost everything in an organism. If it's getting done, if somebody's asking you what does this, default to proteins, because proteins do everything cool in an organism. It's always a protein. They can be used for structure, just like carbohydrates and lipids are used for structure. They can be used for contraction. So when your muscles get smaller on purpose because you want to lift something, that's a protein that's going to do that for you. They can be used for storage. They can be used for defense, like antibodies that are fighting off diseases. They can be used for transport, so they can move things around inside your cells. They can be used for signaling, so they can send a message from one part of your body to another part of your body. And they can be enzymes. And we're going to talk more about enzymes later, a couple units from now. But basically, enzymes just speed up reactions for us, speed up chemical reactions for us. Proteins are made up of carbon, hydrogen, and oxygen, just like carbohydrates and lipids were, but they also have nitrogen. So when we add nitrogen in there, then we've got a protein. Okay? Amino acids are the building blocks of proteins. So just like monosaccharides are the building blocks of our carbohydrates, amino acids are the building blocks of proteins. So an amino acid structure has this central carbon. It has a carboxyl group. So again, that C, double bond O, here's an O, and then this one is a negative. What would we expect to be there instead of it being a negative? Right, a hydrogen. We expect a carboxyl group to be C double bond O and then OH. But since that H is missing, this oxygen has a negative charge because it's got that electron hanging out there. And then we have the amino group. And again, this amino looks a little bit funky, right? Because we normally think of an amino group as being what? NH2. But this isn't NH2, this is NH3, which is why we have a positive charge over here. And then we've got an R group, and the R group changes. So every single amino acid has a central carbon, a hydrogen, a carboxyl group, an amino group. And then just whatever's dangling off of here is what's different, okay? So it's kind of like a Pandora bracelet. A Pandora bracelet starts with that same bracelet and that same funky clasp that a Pandora bracelet uses, and then whatever you attach onto it is going to be different. That's how an amino acid is too. So you've got this basic core, and then just whatever's dangling off of it is the thing that's different. So here's an example of aspartic acid. Central carbon, carboxyl group, amino group, hydrogen, and then here's our varied chain. Again, central carbon, carboxyl group, amino group, hydrogen, and then here's our chain that changes, that varies. So here's glutamic acid, here's aspartic acid. Amino acids have funky names. There are about 22 different amino acids, and I say about because, it depends on who you ask, and it depends on what state you're talking about. If you're talking about ones that you can find just floating around in nature, that's one number. If you're talking about ones that you can eat and your body can use the way they are, that's another one. If you're talking about ones that your body creates once it has eaten other ones, that's a different number. But about 22 amino acids. So again, amino acid plus amino acid, water comes out. What do we call this chemical reaction? Dehydration synthesis. Good. When we get this, what do you think we're going to call this? Yeah, well, it's called a dipeptide. And we call it a dipeptide because that bond there is called a peptide bond. So when you hook an amino acid to another amino acid, what you get is a peptide bond. So we have a dipeptide here. If we hook on more amino acids, what we're going to get is a polypeptide. And a polypeptide is basically what we would call a protein, although there is technically a slight difference between a polypeptide and a protein. Yeah. So the amino acids are connected by dehydration synthesis with special bonds that are called peptide bonds. Proteins have a primary structure, a secondary structure, a tertiary structure, and a quaternary structure. So just like in ecology, when we talked about primary consumers, secondary consumers, tertiary consumers, and quaternary consumers, it's the same thing with proteins where there's primary, secondary, tertiary, and quaternary. The primary structure of a protein is just the order of the amino acids. Hey, we have lysine, proline, methionine, aspartic acid, glutamic acid, tryptophan. Okay, that's the primary structure. The secondary structure is that then that primary structure is going to either bend or it's going to twist up. So we call it an alpha helix. That's if it twists, we have an alpha helix. Or what we call a beta pleated sheet, which is like a zigzag, kind of like a fan. Like if you use a piece of paper to make a fan and you fold it back and forth and then you hold it and you go, ah, my fan. That's the secondary structure. Tertiary structure means that then from that helix or that fan, then it crumples up on itself even more. And then with the quaternary structure, you've got this crumpled up glob, and then another one crumpled up glob, and another one crumpled up glob, let's say, and those three crumpled up globs get together, that's a protein. So when I said there's a difference between a polypeptide and a protein, that's what I'm talking about. So polypeptide, all clumped up tertiary structure, polypeptide, all clumped up tertiary structure, put them together, now we have a quaternary structure, and that's a protein. I know. Okay, proteins rely on shape. If the shape changes, the protein cannot work. As soon as I say protein, the first thing that needs to go in your head is shape. Protein, shape, protein, shape, protein, shape, proteins and shape, proteins and shape, proteins and shape. If a protein changes shape, it cannot work. Proteins have to have the same shape or else they cannot work. Well, they have to have the shape that they're supposed to have. Okay, so every single protein has its own shape according to what its job is. If you change that shape, that's called denaturing or denaturation, okay? And there are three ways you can change the shape of a protein. One of them is that you can change the pH of its environment. You change the pH of the solution it's in, you're going to change the shape. Another way that you can change the shape of a protein is temperature. You change the temperature. Some proteins do really, really well in high temperatures. So if you make them cold, they're going to denature. They're going to change shape. They're going to be bad. Some proteins do really well at sort of human body temperature. If you change the temperature, they're going to do bad. The shape is going to change. They're not going to be able to do their thing. Salt concentration is the last one. So if you change the concentration of salt, you're going to change the shape of the protein. Protein can't do its job. So those three things, changing the pH, changing temperature, changing the salt concentration, that's going to change the shape of the protein. If you change the shape of the protein, it cannot do its job. This is important for you to know. Nucleic acids. The monomers of nucleic acids are nucleotides. They come in three types. There's DNA, which there's only one type of DNA, and then there's RNA, and RNA comes in three types. You don't need to know the difference between DNA and RNA right now. That's all you need to know. And you need to know that nucleic acids hold genetic information. Now, that's an okay definition for you to know right now for the function of nucleic acids. Later on, you're going to learn way more about nucleic acids, and you're going to have to know a way more specific definition of what a nucleic acid does. But for now, that's all you need to know. And for now, that's all I've got for you on biomolecules. Wasn't that so much fun? Woohoo! All right, talk to you soon.